Thank you, welcome. My name is Christina Riemenschneider. I'm a postdoc at the Weizmann Institute of Efrat Schemer's lab. And as you see, my title is like From Chaos to Opportunity, Redirecting the Escape Routes of Breast Cancer Cells. And I forgot to take this. So now I mentioned, I'm really fascinated about the fact that a single cell can generate a multicellular body or human being with about 250 different cell types. We have, like, for example, the skin cell, which is protecting us from the outer environment, or the heart muscle cells, which gives us a beat. But when we look more in detail, then every cell has the same genetic information encoded in the DNA. So what does actually give the cells the character and telling the cells which genes to turn on and to turn off? And there we have a different layer called epigenetics. And epigenetics are small modifications either directly on the DNA or on the histones, which is actually the packaging proteins where the DNA is wrapped around. And these small proteins have a lot of small tails where you can have multiple different modifications. And these modifications can drive now that the chromatin, the DNA, is closer together. So genes which are in this region are turned off, or it is more loose, and then genes in this region are turned on. So it's like a switching, turn on and off for every cell in a specific way. And there are certain, we call it epigenetic regulators, which are setting these small modifications, or they erase them, or they can read them. So, but what is more important is that the cells in their character keep in this state. So you need to lock them in the specific state so that my skin set is not randomly reprogramming back to be a muscle cell. So we don't want that. And this is also doing this epigenetic landscape. It's like locking the cells in a certain state. We call it epigenetic barrier. But what has this to do with cancer? So actually in 50% of human cancer, we find mutations in these epigenetic modifiers. And this leads that this epigenetic barrier is now lowered down, and the cells are able to switch from one state, or from one character, to another one. So this is what we call plasticity. So they are really able to change their behavior. And this leads then that you have a single tumor, and it's composed of cells from different characters. And this has a huge clinical implication, because the cells are now able to switch their state, the character, and they can cope with the environment. And often this leads to drug resistance. So when you use chemotherapy, you can kill 99% of the cells, but some of the cells switch and they are resistant and they stay in the body. And this can then lead to a relapse after some years of surgery, chemotherapy. The cells stay in the body, and afterwards they can spread out to lead to metastasis. So we are working with a model. So we are working with breast cancer because it's one of the causing death or in women for cancer. And actually these breast cancer cells have this high heterogeneity. So and then we call this plasticity because the cells can switch from one cell state to another one. And as an example, I want to give you one of the switching. So the cells can be in a more epithelial state where the cells are in the tumor connected to their neighbors, but then they can switch by changing their epigenetics to be more disconnected from the neighbors, and this can then cause some metastasis. And as you can see, it's here that the more epithelial cells are more drug sensitive, so you are able to treat them better, but when they switch to another state, they will escape from the drug itself, and then these are the cells which stay in the body. But they can also have an intermediate state where they actually have a program which shows genes which are active normally in epithelia or mesenchyma, and they're able to change depending in which microenvironment they are. And as you also see, the mesenchymal cells, they are easier to invade, for example, somewhere else in the body. So, and what we are actually doing, because we know that epigenetic is reversible, we want to address this to better understand the mechanism and either stop the cells to change their behavior and then make them more accessible for the treatments we have, for example, chemotherapy, or we actually change it in a way to switch them back from the mesenchymal state back to an epithelia. But this is only like one example, and there are different axes where they can get other behaviors. So how we do that is we are investigating for every single cell multiple epigenetic marks. So we isolate, for example, tumors we get after surgery, and we are using antibodies, which are small molecules binding to the epigenetics marks of interest. And we have the possibility to investigate for a single cell up to 40 different epigenetic modifications. And not only that, we also are able to use cell identity markers to see how the epigenetics or the landscape is connected to the cell state itself. So, and then we get like a multi-dimensional data, as you can see in this map, and every single dot is a single cell. And then it shows us, do we have different populations which are associated like with a certain epigenetic modification, and if the cells are maybe have a marker which shows us that we can easily treat them, for example, chemotherapy or other therapies. And here's, for example, an example where we actually use breast cancer cell lines, which are actually, we were thinking they are more homogeneous, but we were able to identify different populations. So you can see the blue big population. This is like more a population which is chemotherapy sensitive, but actually we see also a smaller population which is more resistant to chemotherapy. And it's not only that we see this in breast cancer cell lines, we actually see it also in tumors we isolated. So it's like the breast cancer cell lines is a good model to investigate that. So in here you can see, for example, the red marked is that there are specific modifications which are now related to one of the populations. So like in the upper part you see the red one. I will not mention which one it is, but it's not only one histone modification, it's like a complex of different ones. And then with the cell identity markers we actually can see, okay, these cells have the possibility to be more sensitive to chemotherapy or more resistant. And now the idea is to understand this and use inhibitors to now switch the cells, which are less sensitive with chemotherapy, to be in a more sensitive state. So in altogether, this would help us to use the therapies which are already available, for example, the chemotherapy, that they are more efficient. So what I wanted to tell you today is like why we actually care about epigenetics in general. So the great thing is epigenetics is reversible, and is one of the causing hallmarks of transformation in cancer cells. And we have actually targets like therapeutic inhibitors targets, so we can actually target it directly and hopefully then change the intratumor heterogeneity to make the available methods we have more efficient. And therefore, I want to thank you for your attention. I'm open for questions.